Hi everybody, so today we're going to be talking about antidiuretic hormones. So let's go ahead and get started. And the first thing I'm going to say is that usually antidiuretic hormone, a simpler way to put it is just as ADH. So what does this do? It is responsible for an increase in water retention in the kidney. Okay? And it usually is going to do this because it's a drop in blood pressure, which we'll talk about in a little bit. So what it's going to do then is it's going to increase the blood pressure. Okay, so it's going to increase our blood pressure. And like I said, so normally this is going to be released when there's a decrease in blood pressure. But before we get into all that, let's take a look at something here. First thing I want to do is I want to draw a blood vessel here. So there's a blood vessel, right? And then inside the blood vessel, I'm going to have what we call solutes. Right? And these are, for lack of a better term, it's kind of like the solids that are in there. A lot of times we're just talking about sodium, right? So there's my solutes right there. And then what happens is we have water molecules in here. So there's my water molecules, okay? If you notice, I have 3 solutes, I have 3 water molecules. When the number of water molecules equals the number of solutes, that's called an isotonic solution. So we're going to call this an isotonic solution. The number of solutes is equal to the number of water molecules. And this is what ADH is going to try to do, is get this back in balance. So let's say now that you haven't drank water in a while, right? You're a little dehydrated. And so I still would have the same number of solutes, right? There's 3 of them right there. And now let's just say we only have 1 water molecule. Now obviously there's going to be a lot more than this, right? So what's going to happen in this case? Because there's more solutes, we're going to call this a hypertonic solution. So this is going to be hypertonic, right? So I have more solutes in relation to water molecules, right? So I basically have an increase in solutes and a decrease in water. So when you have an imbalance like that, or when we're comparing the amount of solutes to water, what's going to happen is, or what we call that, is plasma osmolality. So in this case, because I have more solutes, we have an increase in plasma osmolality. Okay? The other thing is because in this situation now there's less water, there's going to be less fluid for the heart to pump. And because there's less fluid for the heart to pump, then what's going to happen is the blood pressure is going to go down. So we're going to get a decrease in blood pressure here. So these 2 things here, the plasma osmolality and a decrease in blood pressure, can stimulate the ADH, right? So these can basically cause the release of ADH. So let's take a look at how that happens. But before we do, let's take a look at this right here. I know it looks kind of like an ant. This is actually a nephron in the kidney. So this is an afferent fiber. Blood's going to come through here. It's going to go through the kidney, and fluid's going to come out, and it's going to go into this part of the nephron. This whole thing's called the nephron. So it's going to go through here. So once again, this is going to be the glomerulus. And then if you notice, I have this round part that goes around this. That's going to be called the Bowman's capsule. Okay, and then these 2 combined are what we call the renal. Remember, renal means kidney, right? Or refers to the kidney. And we're going to have the renal corpuscle. Okay, so now here's what's going to happen next. Is after any fluid in here leaves, it's going to go into this part here, which is called the proximal convoluted tubule. It's going to come down to this part here, which we're going to call the loop of Henle. Okay, so there's my loop of Henle, right? Then it's going to come back over here, and we're going to call this the distal convoluted tubule. It's distal because it's furthest away from the glomerulus. So let's take a look at this. I'm just going to move this right over here. And then finally we're going to come to this area right in here. And this area in here is going to be called the collecting duct. So this area right here is the collecting duct. And it's going to collect, or the fluids are going to pass through here. Our water is going to pass through the collecting duct. And then whatever goes in here is going to go down, and it's going to become urine. On the collecting duct, we have cells here. Okay, we have these cells here. And what we're going to do is we're going to look at one of these cells. So I'm going to take one of these cells right here, and I am going to make it larger. All right, so let's make it larger, okay? So we'll come, and this is called a principal cell. That's called the principal cell, okay? We're gonna come back to this in just a minute. So once again, what has happened is we have a decrease in blood pressure, right? We have a decrease in blood pressure and an increase in our plasma osmolality. So I'm gonna draw a brain right here. Let's draw this a little bit bigger. And I'm gonna draw, here's the brain, right? And then up here, on this part up here, what I'm going to have is my pituitary gland. So here's the pituitary gland. This is going to be my posterior pituitary, and that's going to be my anterior pituitary. So this is the posterior pituitary. Right there, okay? Inside the brain, if you recall, we also have an area which we call the hypothalamus. And this is going to be an area that's right above my pituitary gland. So this is going to be my hypothalamus. And then inside my hypothalamus, there is an area called the superoptic nucleus. So I'm going to just draw some nuclei right there, okay? And we're going to call this the superoptic nucleus. Okay, that's the superoptic nucleus. And guess what it's going to make? It's going to make ADH. So, these nuclei actually have axons that come down, and they're going to come into this posterior pituitary in here. On the hypothalamus, we have some receptors. So here's going to be a receptor right here, right? And we're going to call this one here an osmoreceptor. And this receptor is going to become activated to that increase in plasma osmolality. Okay, that's going to become activated because of that. So this is going to sense when the blood comes by and it senses that there's more solids in there, this is going to be stimulated. When it becomes stimulated, it's going to send signals over to the superoptic nucleus. All right. Also on here, we're going to have another receptor. I'm just going to draw this one down here. Right, we'll have another receptor in here. And when I get a decrease in blood pressure, when you get a decrease in blood pressure, what's going to happen is the kidneys are going to release a hormone called angiotensin II. Okay, it's going to release angiotensin II. And so what's going to happen now is this angiotensin II is going to come up and it's going to activate this receptor here, right? This is an angiotensin receptor. And then what it's going to do is send signals also to these superoptic nuclei. So when it does that, what the superoptic nuclei are going to do is they are going to make ADH. The ADH is going to travel down these axons and then be stored here in the posterior pituitary. And like I said, so when these become activated, then it's going to release the ADH into the bloodstream. So there's the ADH. Now, over here on the principal cell. On the principal cell, we have a receptor. There's the receptor. And we're going to call this a V2 receptor. And so the ADH now is going to come and it's going to activate this V2 receptor, right? So there it is. There's my ADH in the receptor. And when that occurs, what's going to happen now is it's going to stimulate a protein. It's called a GS protein. The GS protein actually has something attached to it that is called GDP. Right? So here, I actually went over all this in the hormone receptor video, but this is called GDP. So when this becomes activated, right? So I'm going to put a little plus sign for activated. What's going to happen is then I'm going to have another molecule called GTP bond to this. Now this is going to be stimulated more, and then this is going to come off, right? That's going to come off, and then this is going to go down to a hormone or to another protein that's in here that we're going to call adenylate cyclase. And then what the adenylate cyclase is going to start doing is it's going to start taking ATP and convert it into cyclic AMP, so that this is going to lose some phosphates. The cyclic AMP then is going to go over to a protein called pKa, phosphokinase A. The phosphokinase A then is going to come up to the nucleus, and it's going to stimulate the nucleus. So here's the nucleus right there. And it's going to make the nucleus make, and here's my DNA, it's going to make the nucleus make messenger RNA. The messenger RNA is going to go over to the ribosome. And then what's going to happen is we are going to end up with some proteins. Okay, we are going to end up with some proteins. And we're going to call these proteins aquaporin 2. So these are aquaporin 2 proteins, okay? I'm going to draw a few more of these. Let's draw some more of these. Now, really quick before we talk about those, on the cell membrane of these principal cells, we actually have other proteins called aquaporin 3 and 4. So this is going to be aquaporin 3 and 4 that are going to be on this membrane. So I have aquaporin 2, 3, and 4. Where's aquaporin 1? It was all the way over here at this proximal convoluted tubules where those can be found. So then what's going to happen is this pKa, this is going to actually go over to the Golgi apparatus, which is going to put this in vesicles, right? And then what this is going to do is this is going to cause phosphorylation, which will cause this to fuse to this membrane. So this is going to come over and fuse to this membrane. Now remember, we're just talking about one little cell in here. So I'm going to get my aquaporin 2s now in here. Okay, I'm going to actually, well, I'll draw them here. Right, so there's my aquaporin 2. I'm going to put them in here just as little lines. Right, actually I'll just draw one. So here's what's going to happen, is now water can pass through there. So the water that's coming down can now be reabsorbed and pass into here. So here comes the water into here. Now it comes into the cell, right? And it can go out through an aquaporin 2, right? It can go out through one of these, I'm sorry, an aquaporin 3 or 4. It can go out now. Sitting out here is going to be a blood vessel. You're going to have blood vessels. So now, let's say we had it, like we said, we had our hypertonic solution. So I drew 3 solutes in there, right? Now this is going to, and we had one water before, right? We said this was hypertonic. Now what's going to happen is water molecules are going to come in here and it's going to balance this out. And because I've added more water now, the heart has to push that water. And because the heart has to push that water, the blood pressure is going to go up. So I've increased the amount of water in there, which is going to lead to an increase in blood pressure, okay? The other thing that's going to happen too is on here, we actually are going to have some other receptors that are going to be on our blood vessel, right? And the ADH can also bond to here. And when the ADH bonds to there, what it can do is it can actually cause the blood vessel to become smaller in size, right? And we call that vasoconstriction. So the ADH can bond to, these are actually called V1 receptors, right? So they can bond to V1 receptors that are on blood vessels. And what that will do is it'll cause vasoconstriction, right? And if you remember from physics, what we said is if we decrease the volume, right? I'm making this smaller, so I'm decreasing the volume. If we decrease the volume, it will lead to an increase in pressure. So now the blood pressure is going to go up even higher, right? Which is an increase in blood pressure. Now remember, we're trying to get low blood pressure up to where it belongs, right? We don't want the blood pressure to go too high, but that's not going to do this, okay? So the aquaporin 2s will actually only be activated when we get a increase in plasma molality, right? Or a decrease in pressure. That's the only time we'll get these, right? Once that's over, these will be taken off. All right, so that's it for antidiuretic hormone. I hope you liked the video. If you did, please hit the like and subscribe button, and we will catch you next time. Thanks you so much for watching.